Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
It's my pleasure to introduce today Professor Tanya Berger-Wolf from the University of Illinois at Chicago, and she'll be talking about the social life of zebras, which rather curiously and by complete random chance is scheduled 30 minutes after another talk on zebras, which I hope you had the chance to catch, by Pei Zhang of Princeton University, talking about ZebraNet. It is a complete random coincidence, but the work that you'll be hearing presented today is, I believe, also related to ZebraNet, and I'm sure that will become clear. So,
okay, thank you. Well, I'll let the. Can you hear me? Actually, okay. I'll let the professional movie makers to start the intro and explain the biology behind and motivation and what it has to do with zebras. So, and then I'll tell you about the computer science behind it.
Thousands of years ago, numerous species of zebras roamed from the Mediterranean Sea to the Cape of Good Hope. Today, 3 species remain, and this one, the Grevy zebra, is fighting hard to survive. But Dr. Dan Rubenstein is fighting hard too, in a race to understand why the Grevy zebra is in such trouble. And what he's discovering might well give this beautiful animal one more chance. Zebras and their wild horse cousins are different from most mammals. On maturity, the youngsters, both male and female, leave home and join completely unrelated groups. So mature zebra groups are made up of complete strangers who actually get along pretty well. Long ago, they discovered communal living has its advantages. Today, this is how most of the world thinks of zebras, the vast herds of plain zebras, 200,000 strong, who, with the wildebeest, follow the lush grassland along the great Serengeti migrations. For Dr. Dan Rubenstein, a behavioral ecologist from Princeton University, the puzzle is getting to grips with what makes animal societies tick.
And we're trying to help him and provide the tools to figure out what is it in the social interactions mainly of the 2 species, the plain zebras that is thriving and the Grevy zebras that is endangered, that are really different, similar in everything but their social interactions, their social structure, that makes such a big difference in their survival outlook. And the Princeton part is not coincidental. It is part of the ZebraNet project, and I'll tell you more about it that you may have heard. But clearly, zebras are not the only animals who interact socially, and the interactions have implications on their survival outlook and various aspects of their life. Social interactions and the implications of those come up in many different contexts, for example, in disease modeling, in cultural information transmission and communication through email and blog space and so on. This is the infamous now Enron email network that was subpoenaed when Enron had its trouble. Intelligence and surveillance, this is the terrorist network or people involved in the September 11th attack and various connections between them. Business management and conservation biology and behavioral ecology, which is dear to my heart and provides motivation for my work. And there's some work has been done there recently, not only on zebras, but on guppies, buffalo, and dolphins. But we'll show zebras as an example. So what is it that we're talking about? We're talking about social networks. Typically, a social network is a set of individuals that we represent using nodes and interactions between those individuals that were observed at any point during the observation period are represented as edges between those nodes, and we may add a weight to these edges that represent either frequency or probability of interaction over time. The problem with this particular view of social interactions, that it's actually static, that there may have been many dynamic scenarios that gave rise to exactly this picture. For example, here are these 6 individuals interacting over a period of 5 time steps. At every time step, we have 2 individuals interacting, 1, 2, 2, 3, 3, 4, 4, 5, and 5, 6. And this will correspond to this network where the frequency of interaction is 1/5, only 1 out of the 5 time steps. Here's another dynamic scenario, another scenario that could have happened and is represented by exactly the same static social network, where again only 2 individuals are interacting in any given time step, but they're interacting in a different order. Those interactions happen in different order. Here's another one with the same frequency of interactions. Everybody pokes their neighbor to the right or sends an email to the neighbor to the right. And besides the fact that these are all different, you might say, so what? Well, what if we're trying to answer questions, for example, who are the most influential people, or who are the ones who are most likely to infect in a disease scenario, disease spread scenario, most individuals in this little population of 6? And if we look at the first example, then individuals 1 or 2 are the ones that will spread over the 5 time steps to all other individuals in the network. Versus in the second scenario, 2 doesn't interact with anybody until the very last time step. So it will never actually reach individuals 4, 5, and 6, if that's where the timeline ends. And in this scenario, these are complete strangers that by accident, by chance, probably happen to be together in this very first time step, and they only can affect their immediate neighbors at the very first time step and never meet again. So if we are going to ask questions like this, then we really need to look explicitly at the timeline of the interactions in this population so we get more accurate information, and moreover, and more importantly, get a chance to answering these questions about the populations and their social interactions. How do diseases and information spread through population? Who are the individuals that are critical in one way or another, for example, leaders or interaction facilitators, or the ones who, and how do they emerge? How do social structures, such as communities, for example, change with outside circumstances, conference deadline or project deadline, or presence of a lion in the area? And how stable are these structures? And are there any recurring ones? So we, of course, simplify a little bit what's going on, but we look at an explicitly dynamic network, and the way we think of it is we have a snapshot of the population at some time step. Here are all the individuals. And we turn away. They do whatever they do, and we take another snapshot. For the purpose of the current talk, for now, we consider spatial proximity. Individuals that are close together are interacting. Okay? So if this is the picture that we see, here are the interacting individuals. Okay? These A, F, C, D are interacting, and these are by themselves. So they're not they're interacting with themselves. So we can take a series of snapshots like that and build this social network of the population. And the way we do take those snapshots, or will in the future, is using the ZebraNet colors that you may have heard earlier today. These are exactly the ZebraNet colors. This is a test on a horse farm done in New Jersey, and that's on an actual zebra, and it looks just like another stripe there. So, and this is the hero who is saving the zebras, Dan Rubinstein from Princeton, who was Margaret Martinez's collaborator, whose student graduate student you may have heard the talking about ZebraNet today. So here's how it all comes together. So the ZebraNet colors, or some equivalent of those, will help us collect this network, and we have some prototypes already of taking these snapshots. And so we get a network like this. This is the aggregate view. The difference for now, most of this network is collected from visual observations, visual scans, not actual GPS colors. We don't have enough of those. This is a static aggregate representation of this network of gravy zebras, of interactions over a period of 3 months. And this is sort of the dynamic view of how it unfolded. From time goes from top to bottom, and each line is the snapshot of individuals interacting. These are individuals that were close together, all close together at the same time. So these two were close together, and this whole set was close together. So because it's not GPS colors for now, we have individuals that are not observed at all times, but we hope to have a more complete view through those colors. Okay? So
that was all from human observers.
right, so there is a fixed loop. There is a fixed loop in this big area in Kenya that they're driving every other day, I think. And it goes through most of the area, and they sort of check the individuals. They see the individuals there, the groups. They check GPS location of each group that they see, or each individual that they see. And there are graduate students who can identify a zebra by its stripes in under 4 seconds flat. So we know exactly who each one of these are. They also take the demographic status, such as female, male, age, and so on. So they know exactly who these are. Okay. So we have, this is where we're going. We will have GPS colors. Now the project funded by NSF, and we'll have information from those. We're using whatever clustering approaches we can identify the groups or infer the network for one snapshot, and then that's one time step. Okay? That's the one time step. We do the same thing for the next time step. So we may observe maybe these two individuals are coming together in the next time snapshot. One of these yellows starts walking away, then joins those orange ones in the third time snapshot. Then one of the green ones joins the yellow ones, and so on. And so when we have a time series of any kind of information, the very first question that we ask typically is, what's next? Can you predict what's going to happen, how this whole snapshot is going to look in the next time steps? And so this is work that is one of my graduate students, Mayank Lahiri, and I are doing. Given a time series of these snapshots, can we predict, or what can we predict of how the network is going to look in the future? So if we can predict well, then we actually know the model of these interactions. So we basically have learned why these interactions happen and what drives those interactions. We can understand the behavior and the social dynamic. We can play the what-if scenario. On the other hand, if we have been predicting well for a while, and then suddenly we don't, then something has changed. Something fundamental has changed in the social dynamic of this population. So it's a tool for anomaly detection. So we ask the question of, given a time series of networks or a fixed set of individuals, when will a previously seen interaction occur in the future? And we want, of course, accurate, adaptive, generic, transparent, fast, shiny, sparkling, green, whatever algorithm. There are many related things out there. For example, there are many generative models of networks that from random graphs to small world and preferential attachment networks that have the model. So if your network actually behaves according to that model, then you, of course, can predict what the network will look in the future. So they generate the network. There's also work on link prediction, which asks a slightly different question of whether a previously unseen link occur sometime in the future. Okay. So if we can do both this and this accurately and well and with good recall, then we can predict the entire network. So we're focusing on the ones that were seen, and we want to know exactly when they will occur again. Okay, and there are, of course, complex realistic models that are fine-tuned for a specific population, for a specific purpose, such as TransSims, the transportation model of Portland, or EpiSims, and so on. Okay, so here's a toy example. We have this time series of interactions of this networks on 3 individuals, population of 3 individuals, and they're interacting in this time steps 1, 2, 3, and we can record this information, and because we're assuming that there is actually dependence between different interactions. Not only that every interaction occurs independently of everything else, but there's a dependence between all the interactions that are happening. So we're recording not the occurrence of interaction, but the delay between any pair of interactions. Okay? And I'm skipping the independent model because it doesn't work. So we have this matrix of edges by edges. And in the entry AC by BC, we're recording all the delays, temporal delays, with which BC has occurred after the occurrence of AC. So for example, AC occurs in the first time step, and then BC occurs in time steps 2 with the delay 1, and in time step 3 with the delay 2, after the last occurrence of AC. Okay? So then what this matrix is doing is recording the basically the random variables, distribution of the random variables of the delay between any pair of edges. So the delays are random variables, and this network records their distribution, discrete distribution. Okay, if that's the case, okay, this is what every entry looks like after some period of time. We can convert it to the frequency of any particular delay. And for discrete distributions, the maximum likelihood estimate of that distribution is the mode of the distribution. So it's just the largest frequency value. And we can predict maximum likelihood. We can build a maximum likelihood predictor by just using the mode of the distribution at every point. So if we were interested in answering the question of when will BC occur next after AC has occurred, then once we see AC occurring, we say, okay, well, what's the current mode of the distribution, the highest value? It's this, so 3 time steps later, BC is most likely to occur. Okay? Not only that, we also have the conditional probability of that prediction, which is the probability of BC occurring 3 time steps after AC is the mode of the delay 3 divided by the number of occurrences of AC so far. Okay, so we transform basically this discrete distribution matrix to this maximum likelihood matrix with confidence. So, are we done? No. The problem is we have the assumption that every pair of edges is independent of every other pair of edges. Because, okay, we have this, that the maximum likelihood predictor here is delay 1 for AB, but there is also if BC has occurred. But there is also delay 3 if AB has occurred, and then there is the delay 2 if AC has occurred. So there is various maximum likelihood predictors with their own probabilities, and each probability was computed independently, okay, for each pair. So there is the assumption of independence of each pair. There is also a lot of noise that we're tracking, and if we're talking about even the population of these plain zebras, which is about 200 individuals, there's about 40,000 interactions. So we're building a 40,000 by 40,000 matrix, which we're tracking over time, over whatever many time steps. So it's completely intractable, really. So what do we do? we've switched to, instead of edges, we switched to frequent subgraphs. So not only any frequent subgraph, but frequent closed subgraphs, and I'll explain what it is. So if we have a series of any graphs, of any nodes and edges, objects, then we have a frequent subgraph, it's part of this graph, that occurs sufficiently many times. And that's a threshold parameter. So say twice, if something occurs twice in this series of graphs, then it's frequent. So here are some frequent subgraphs. We have this red subgraph is frequent. This is a frequent subgraph, occurs at least twice. But also this is a frequent subgraph. Okay? It occurs also twice. So looking at just frequent subgraph is a problem because any subgraph of subgraph of a frequent subgraph is frequent in itself. So you want maximal frequent subgraphs. You want the maximality in structure. So that's the red one. The red one is the only maximally frequent subgraph here. But even though green is a subgraph of the red, it occurs at other time steps where red does not occur. Okay? So it's temporarily maximal. And that's the closed subgraphs. So closed subgraphs are frequent subgraphs that are either structurally or temporally maximal. So in this particular example, there are two of them, and you can track them very, very efficiently. You can extract them and track them efficiently using frequent item sets if you're interested how to do it. So what it does, though, it filters out insignificant edges. It also aids tractability for large networks, but also intuitively, if you have a frequent subgraph, then it's something that's probably more likely to be predictable and also interesting, although it can be trivial. And the frequent closed subgraphs capture the distinct patterns of interactions. And so now we go from this matrix of maximum likelihood delay for edges to maximum likelihood delay for frequent subgraphs. And with a little bit more of manipulation of probabilities, you can actually extract maximum likelihood delay predictor for this structure. And so we then go ahead and test it on real networks. We take a time series of these networks, take the first time step, the first 20% of the time steps as training ones for frequent subgraphs to extract frequent subgraphs, and then we start predicting from then on. We tried it on many different networks. I'll show examples of 4. This plain zebras, which is a population of about 1,000 individuals over 200 time steps. Here are the rest of the stats. We have the infamous Enron, which is nodes are individuals, and an edge is there if the email was sent from, so individual email addresses, and an edge is there if an email was sent from one address to another over a 3-year period. And then we created 2 completely artificial ones. This is New York Stock Exchange. Each node is a stock ticker, and an edge between 2 nodes exists if their closing prices, daily closing prices, highly correlated over either a sliding window of 30 days or 5 days. So we created 2 different instantiations of this network. So before I get too excited, it's a completely meaningless financially network because closing prices are not predictive of anything, actually. So yeah, but this allows us to test this predicting algorithm, and here's what we find. So here's Enron and zebras, and this is the 2 stock networks. This is the x-axis is the minimum support of the frequent subgraph mining. So what do we consider frequent? Of the first 20% of the time series, if a subgraph shows up 5% of those, then we consider it frequent. 10%, 15%, and so on. So at 15% support, so something that shows up at least 15% of the first training part, we can predict then the rest of the time series with about 60 or 70% accuracy for zebras and Enron. And here we need, so clearly New York Stock Exchange is much, much noisier network than these ones, than actual social networks. And here we need at least 40% support, which means that we're actually predicting pretty trivial structures that are almost always there. Okay, yes?
Why do these tests stop at roughly 15%?
Because then it starts dropping off. You're picking up too much noise. So it improves a little bit, and then once you start tracking more than here, more than 15%, so what you consider frequent is more than 15%, there are two things happening. You have very, very few of these. There are almost none in this case after 15%. There's no subgraph. And the other thing, those that are there are actually not predictive anymore. Yeah, so you're overfitting. Yeah. Okay. And random prediction gives you about 10% accuracy, just as a baseline.
What's the prediction test? Is it dependent on the frequent subgraphs that you find?
The prediction test?
Yeah, the task that you're trying to predict is based on the frequent subgraphs that you
So
find.
we're right. So we're predicting this frequent subgraphs, and once we predict for every time step, we predict what we predict, and then only for that part we say, okay, is it there or is it not there?
Non-appearance of
Right.
one of these subgraphs that you identify.
Of the edges. We're actually tracking the edges. We're predicting the subgraphs, but the accuracy is based on the edges. How much of the edges we're actually predicting correctly. Yes?
I'm not sure I understood that, but let me rephrase the question, which might get to that. So when you're predicting, so supposing in the Enron case, they suddenly fired half the company. You won't have those people participating in that.
Exactly.
So your model so far assumes that the world is, relatively stable and,
Well, as any model, if you learn the model, the world is stable. And if suddenly you're not doing well, your model is not predictive, then there is an anomaly and you should go
so
to
your Enron data you said was across 3 years.
Yes.
Organizations could be real. So it's kind of you're shooting yourself in the foot by trying to predict across time.
Exactly. Yeah, so it's actually an adaptive algorithm. It keeps on updating that distribution. The problem is that for this implementation, we didn't update what a frequent subgraph. Now we have an implementation which actually keeps on changing the set of frequent subgraphs as it moves along. So it's an online algorithm. The reason we cannot call it streaming is because the memory constraints are more than streaming algorithms by definition require. But yeah, it has to be adaptive and online. So you're right.
So your figures should be better now.
Our figures go to 80%. It seems we cannot break the 80% barrier, and we compared it also, and I can show you offline, compared to neural nets, SVM, anything else you can throw at it. We're doing way, way better than any neural net and faster. We're doing comparably, for some data sets, a little bit worse, and we're talking like less than 1% worse or better than SVM. But the advantages is it's not clear how you set up SVM for this. It's kind of doesn't always work. You can't set it up for all. And we actually can extract actual humanly understandable temporal patterns from this. If these interactions happen now, then with high probability, these set of interactions will happen 3 time steps later, which you don't get from SVM. Yeah. The part that is bad about all of this was that we're predicting a very small part of the network. So the recall is really bad. But arguably it may or may not be important or a goal or an objective in all of this to have a high recall because there is a lot of noise. So there are a lot of interactions are noisy. What we're predicting highly accurately, reasonably accurately, is the part which is the core, the pattern, the stable pattern of interactions. Okay. Okay. So that's the prediction. The other interesting part, once you know the prediction, you've learned the model, you know how everything works, you can identify anomalies, detect anomalies, and all other patterns. And by the way, in another development of this, we now look at patterns as recurring patterns also. So there is an offshoot where I can talk to you offline. So the other thing is patterns of interactions are maybe recurring and persistent sets of individuals that are interacting, and that's communities in this population. So that's work with Chen Tantipathananam, who is another one of my graduate students, and David Kempe from University of Southern California. So zebras are very, very fluid species. They're called fission fusion species, like many other equids, horses, that they have groups. The populations are made of groups that constantly form and break and reform. And when you look at it, it's just complete, seems to be complete remixing. But is there any cohesive part, any coherent part to all of this breaking and forming and reforming? And do these groups have an effect on the genetic diversity through who is breeding with whom? And at what time, who is in group with whom, and vice versa. We heard in the movie that these social interactions, these groups are made of completely unrelated individuals, or are they? Do they form these groups because they're unrelated and they avoid their close relatives, or is there some influence of the genetics on the group formation, and what kind? So in general, we want to identify these cohesive groups, but we want also to allow a fluid structure and individuals should be able to be part of a cohesive group even if they're not observed with other individuals in that group at some particular moment. So these are dynamic communities. So from social sciences, cohesive communities are cohesive subgroups, communities, are subsets of actors among whom there are relatively strong, direct, intense, frequent, or positive ties. So in computer science words, that means community is a cluster, which means community is a set of individuals that interacts among themselves more or more intensely, or more frequently, or more consecutively, or more something than they interact with other individuals that are not part of the community. So dynamic community is a dynamic cluster where we want this consecutive or some temporal aspect of it when we say interact more with each other than interact with members of outside of a community. So the computational approaches, most of them are now focused on this aggregate view of the network to identify communities. And there are several. There is based on centrality and betweenness of individuals or interactions, started mainly by, in computational world by Gervan and Newman's paper in 2001. There is the purely graph theoretic approaches of correlation clustering, and there's also the approach of overlapping cliques, or in general this whole direction of dense areas of the network. So we focus specifically on dynamic communities, and so the thing is, so here's the static view. Remember this network? All static or aggregate community identification approaches will identify this as one community, and then they'll do different things. Well, clearly this is another community, and then they'll do different things with these individuals. Well, here's the dynamic network, the dynamic view. So, and I'll show you sort of what we do with this. So the model, the dynamic communities model, we have this, again, time steps. We consider them already within each time step. So time is discrete. It's been given to us. Also, these groups, the group gatherings within each time step, the clustering within each time step is given to us. We can talk about how, but for now, assume that it's given to us. So it's an affiliation network, not actual social network within each time step. So it's meetings, maybe conferences or different kinds of meetings. Okay. So there is, each meeting we assume represents some community. There is this particular meeting of people who are maybe interested in social network analysis. There is some other meeting going on somewhere of people who are interested in golf on yellow grass. I don't know. But there are different meetings, and so meetings, these boxes, represent some community. There's also individuals that can show up at different meetings who have their own community affiliation. So here's this yellow individual that went off by himself and did not join the yellow meeting at the second time step. And these two individuals, the green and the blue, may be joined together in a blue meeting, and the blue one convinced the green ones to join his community affiliation. So both meetings and communities, meetings and individuals have community affiliation and community representation. Okay. And even though some individuals can show up at meetings of other communities, they may or may not necessarily change their personal affiliation. That's the model. So here's the data. We get it like this. And we represent it as, for each time step, the set of nodes that correspond to individuals and the set of nodes that correspond to these meetings, the groups for that time steps. And we have an edge from each individual to the group node where it was observed for that time step. And we do this for every other time step, and also introduce edges from each individual to itself to enforce consistency, individual consistency. So now the model, we have sort of arbitrary assumptions and necessary assumptions. So constraints, arbitrary constraints. For now, we assume that individuals and group represent exactly one community at any given time. So if we have an individual can have only one loyalty at any given moment, and a group can represent only one community at any given moment. So every node, whether community or individual, is colored only one color. Concurrent groups represent two different communities. If there were one, they would be meeting together. So at every given time step, group nodes will have different colors. Those are sort of arbitrary assumptions, because there are possibly instances where you want to have individual have two community affiliations at a given time step. But for now, that's that. And here are the necessary costs. So we assume parsimony, basically, that there is conservatism, that community affiliations are rare, changes are rare for an individual. So every time an individual does change its community affiliation, we enforce a cost, call it alpha. We charge some cost alpha. Also, individuals tend to show up in meetings of their own community. Okay? And if they don't, if they show up at a wrong meeting, which may happen, but we'll charge them some cost beta 1, or if they don't show up, if a blue individual doesn't show up at a blue meeting, we'll also charge them a little cost. And finally, there is overall parsimony, which we can drop also, and the model goes through just fine, that there are few communities overall for each individual. Okay, with these assumptions, we can formulate an optimization problem, which is minimum community interpretation, that for a given setting of these costs, we basically find a coloring of groups and the individual nodes that minimizes the overall cost for the network. So the group vertex coloring is the community structure, and the color of individual nodes is the affiliation sequence for that individual. For those interested in more theoretical aspects, it's NP-complete, it's APX hard, and they're great heuristics, very fast heuristics. So with these heuristics, what do we get? So remember these network. So all static community identification algorithms will identify this as one community. What's going on in the dynamic sense? This is the result of this community identification algorithm, the dynamic community identification algorithm run on this network. So the boxes are group nodes, and the circles are individual nodes, and the color represents to community. So you see there is this little community that blipped was observed just twice, which is this for individuals. Overall, there's one big blue community that corresponds to this mess. And there are other peripheral individuals. There are two interesting ones. There is this green individual. It's a male that was together with other female, just single female, a bachelor. So all these blue ones are females. This bachelor tried to, it's this one, tried to come over, join the females, take over the harem. They kicked him out. He went back to being a single bachelor. So he didn't actually change his community affiliation of being a single bachelor, even though he showed up in this group for a couple of time steps. On the other hand, this individual, which is this female, who also was in a very loose affiliation with these other 4 females, actually joined this group, this blue group of females, and stayed and became member of their community. And our algorithm picks it up. Okay.
I thought your algorithm would give a single color to each individual.
At any given time step. At any given time step. Okay.
So the line connects.
Right. So the line connects individual to itself. Yes. Right. So we allow these community affiliations. The algorithm basically minimizes, and it has to figure out if it doesn't change color, then we will charge this individual this cost of being different from the group it's in for all of these time steps. Or we'll charge once the, sorry, it's this one, the switch, the community switch cost.
And with the bachelor that you're talking about first, it knows that...
The minimum, the optimal, right. It's cheaper to just be an odd one out.
Because that's the parsimony constraint you mentioned, that you don't want to change things too many times.
Well, because we have the cost right now of the loyalty of being the odd one out in a group, in not your own group, or actually paying for changing your community affiliation. Okay, so it could have changed to blue here, and then could have changed back to green. So they would have been twice versus being twice the different one. And so overall, it depends both on the cost settings and on the overall global optimum. Mm-hmm.
So when
The clustering within each one?
how do you decide the groups? I mean, so
For now, because they come from visual scans, they see these groups. The biologists see these groups, so it's easy. They see a group being there, they take GPS location, they write down the individuals. When you have GPS colors, it's going to be a lot harder.
Yeah, there I could see a core blue group versus some, people who come in and
Right,
out.
because they saw them at different locations at the same time. Okay, they're literally in different parts of the landscape. Yes. But you need...
That can change your interpretations.
Well, for these particular ones, I mean, when you look visually, we trust the biologists to do it. Let's put it this way. Once you have a, automatic GPS data coming in, it's a much harder question, and there are other options out there. So from GraphScope, which most recently does this information minimizing clustering, or one option, or just straightforward clustering for every time step. Yes.
So in that case where it was cheaper for that individual to maintain the individual coloration and join the other group, how do you deal with the fact that cost might have been more cost effective to change affiliation, because I see that individual was not observed for many of these time periods?
Right. So we do have, remember, the cost of being absent from your community, and that's where it comes in. That's the,
okay, so
if it's not observed, but yet there is so what is it? Right. So here, that individual, this one, even though it's a member of the red community, it's not observed at this time step. So there is a cost of that individual being absent from the red community meeting.
okay, so we're imposing that cost both for not observed as we impose for observed but not in the community.
Right, so if all individuals are observed at all, this is these two costs. If all individuals are observed at all times, then we don't need both of them. Because if it's the orange individual is present in the blue meeting, then it's absent from the orange meeting at that time. So this cost is specifically for unobserved individuals, basically.
Okay.
Yeah.
And that cost is lower. Beta 2 is less than beta 1.
Yes.
Could you go to the picture where you see the dynamic that? When you run the optimization, is it aware of all time steps to undertake that?
You can do it 2 ways. So, and actually we've tried it more than 2 ways. We've tried it in many different ways. So you can do a sliding window. You can do the entire network global optimum, which actually is not good. You can do sort of this flow-based approach, which it's reasonably global, but it's aware of the forward direction. So there is this basically greedy local thing. And yes, the greedy local heuristic for a reasonably global optimum is the best thing. So it should be aware of enough time steps to optimize, but the decisions are reasonably local.
Okay, so the next question is going to be a tricky one, even to formulate. How sensitive are you to the fact that you may have adjacency in clusters? So it's a group.
We don't know yet.
Okay. So do you have some notion of like a radial distribution function of your?
We're playing around with it, but it's a big question, yes, and we're trying to figure it out because you need to control for a lot of things to isolate that particular one. And
okay. Have you done
it's an important one.
any modelling where you've taken? So I come from a molecular modelling background where one could simulate a whole load of behaviours
yes.
and track and remove some of the
yes.
issues that you've got from some of the field experiments.
Absolutely. So, and actually you can come up with a very, very simple Markovian model of community changes. So you fix which groups a priori, here are the groups for these communities, here are all your communities, and here are all your individuals. Now, ready, set, go. The only thing that you say is that the probability of, so at every time step, an individual decides what community it's going to join and what group it's going to join for that time step. Okay, and the only constraint that we impose is that the probability of switching a community is less than the probability of staying within the same community, and the probability of of joining a group which is not part of your community is less than the probability of joining a group that is part of your community. So that's it. So then that Markovian model, if you do the math, this particular optimization problem is the maximum likelihood estimate for that model. And if you want to take it even further, we can talk maybe offline about different group selection model, and the tag model for group selection, if you're familiar, or if not, I can talk about it, is you can interpret this Markovian model as a tag model for group selection. So it has a reasonably good foundation. Okay. So the fun part is also that you can compare, using that, you can not only infer the structure for one population, you can compare 2 populations. And this is the Grevy zebras, 28 individuals over, I forgot how many time steps, some number of time steps. This is onagers, which is wild asses in India, a population, also donkeys, equids. Very, very, very similar social structure. Both harems, both species are harem species, many females, one in very loose harems. Males establish territories, females decide or not decide to join as a group. Groups of females are very fluid. Population of 29 individuals over the same number of time steps with the same set of parameters for alpha, beta, gamma. Actually, we dropped gamma for this, so for alpha and beta costs. Very different picture. So here's the static network. It looks actually pretty similar. It's the layout, unfortunately. We need to change the layout. Very similar to the other one. It's this core of everybody connected to everybody, and then some peripheral nodes that are loosely connected to these individuals. And when you look at the dynamic community structure inferred from this, it's very different. Clearly, the structure here is much more coherent. It's much more, cohesive. Much more cohesive. It's more persistent, even though at every given time step there is kind of mixing of who's with whom. Here, there is literally no loyalty to community. Individuals are observed in all kinds of groups that are basically not their own color. So this is a much less cohesive structure. There is no persistent community structure overall. You need to enforce to make the costs really, really high to make this reasonably monochromatic, which means that comparing those two populations, there is very little individual loyalty to communities, so individuals do not choose their affiliations based on community loyalty, but rather opportunistically at every given moment.
How does the left-hand graph look if you take the right-hand optimization?
It's the same. They're the same costs. That's the whole thing.
But you said you have to change the parameters to optimize for
Oh,
the right-hand.
it's monochromatic way before that. It's way before that. Yeah. So that literally tells you that there is no, that the interactions here are not driven by some community affiliation. So here they are. They actually are probably this core of females that keep on coming back together over and over again, versus here there are very few, I mean, there is this, if you track these blue individuals, they're actually not, for the most part, I mean, they're part of the same loose group, and that's the only loose group that actually very short-lived that happens there, and that's the case. I mean, biologists, this is for the first time they actually allowed to quantify this, something that they knew before, but we can provide a number now. This is more cohesive than this, the structures here are more persistent than here, and the lifespan of a structure is literally about 2 weeks, about this much. Okay, here. And the interactions are not driven by sort of liking of your friends over a long period of time, but by what's happening right now. Okay. So, very briefly, running out of time, I'll also tell you a little bit what we're doing about the spreading behavior in these networks. So this is work with another one of my graduate student, Habiba. So you have these networks, not only there are interactions that are happening in these populations, but things are happening, spreading over these interactions, such as diseases, behavior, opinions in human networks, and product liking and marketing and stuff like that. So for zebras, the question becomes, for example, if there is a line in the area, there are several false starts of trying to run away in different directions, and finally they're all following one or two zebra that's running away in the same, and all of them are going in the same direction. And you can literally see, like, one starts, nobody follows. Okay, I'll come back. You know, another starts, nobody follows, and then finally. And what turns out biologists think, their hypothesis is that who they're going to follow depends on who they have been interacting recently, reasonably recently, who they consider worth following to begin with, who they know, who they consider an authority within their old close circle. So they're not going to follow anybody. So we need to identify how things, there's the community part comes in. But so it means that their influence happens over social interactions, spreads over social interactions. But there's also disease spread in these populations and so on. So we have these interactions that were observed. Sorry. We have these interactions, and something starts spreading in this population, and so the color orange through interactions, the other individuals have become orange, and at the end, here are the orange individuals. And the first question is, okay, well, who is the most influential? Who is the individual that if we starts orange here, there will be most orange individuals at the end, the largest number of orange individuals at the end? Is this the one? Maybe if we started from somebody else, it would be, we would have more orange individuals later on. And so you can formalize this question using mathematical models of spread that have been developed since 1970s. There are two sort of extremal model. One is independent cascade, which is each the spread happens independently over every interaction, over each interaction between two individuals, independently of what else is going on with these individuals at that moment, and you have this fixed probability of that spread succeeding. So if I tell Simon you should really go and see this movie, his decision of going or not going to see that movie will be independent of who else is telling him to go and see that movie. So that typically is appropriate for disease spread, where each contact has independent probability of succeeding. And the threshold model is, for each individual, there is a certain threshold. When enough of his friends are telling him to go and see that movie, then he'll probably go and see that movie. So each individual has a threshold of influence, and when enough of the friends are bugging him, he'll go see. So that's typically used more for influence spread and opinion spread and behavior spread. And there are mixtures between those. And the general schema is, so some individuals start active or orange in the network, and then through interactions using whatever model, there's more and more, at each time steps, there's more and more active individuals. And for aggregate networks, when the network is that static representation, there is a result of Kempe, Kleinberg, and Tardosh that that the best you can do in some senses, if you want to find a group of influential individuals, is find them one at a time, and the only way we know to do it is through simulations, stochastic simulation of finding one by trying spread from everybody. You know, you find one it results in most spread. You take that individual out and everybody who it affected, and then you do the same thing, and you find your set of most influential individuals. So I'll skip all the setup for the network, for dynamic network. The same thing works for dynamic network. You can do the same sort of greedy approach. You find the most influential individual through simulation, remove it, and find the next one, and so on. And you do pretty well. You find a good set of influential individuals. So the differences, though, so this heuristic works very well. This approximation story works very well. The differences between the static and the dynamic, not only the dynamic, we did a lot of simulations. This is for gravity zebras again, but I can show you other networks. For independent cascade model, the maximum number of individuals that you can reach when you do dynamic simulations is fewer than for static simulations, for the static model. So the static model overestimates how many affected individuals you can have. And the individuals that are identified as influential by the dynamic algorithm are different than those that are identified as influential by the static algorithm. So the static, and clearly dynamic is the truth, right? So the static algorithm misidentifies actually the influential individuals. And this is for independent cascade model, the same thing, the same holds for the linear threshold model, except the static model underestimates the extent of spread. But again, the influential individuals are different between the static and dynamic algorithms. So we are now conducting a large set of experiments on this whole spread, asking different questions about spreaders and blockers, and identifying structural properties of the network that are indicative of good spreaders and good blockers of spreading processes in the network. Because remember, we can identify a set of influential individuals, but we have to do it one at a time through a massive set of stochastic simulations. That's not the way to do it for large networks. So you want some indications. Maybe it's individuals with large degree. Maybe it's those are individuals that are high centrality or something. So it turns out, and again I can talk to you offline, that there is a reasonable set of structural indicators of who are the good spreaders and blockers, but they're different for different kinds of networks. Okay. So hopefully over the last hour I've convinced you that dynamic is really different from static. We really need to develop methods that address the order, concurrency, and the delay of interactions explicitly, which is what makes a dynamic network. We have a few things, but there is a lot more to do, and it's fun. And zebras are better than humans because they don't have ulcers. Well, if you haven't, it's a book by Sapolsky, Robert Sapolsky. It's a great book. Zebras don't have ulcers, as a side note. But they don't have cell phones or emails, so proximity, spatial proximity, is reasonably well corresponds to interactions. There is also no privacy issues, which is a big headache in all of this research. And we also can conduct experiments and validate and test a lot of these things, because when we tell you, okay, this is an influential individual, how do you test that? Well, in zebras, or rather in horses, we're going to conduct experiments on horse farms around the country. When we identify an influential individual or an individual that holds, by our prediction, the social structure together, we can take that individual out and see what happens to the social network. Or we can start running that individual and see if everybody follows. Okay, so we can actually validate a lot of that stuff on zebras, which you cannot really do on humans. So, but clearly whatever we do does apply a lot of it to humans, maybe not harems. And I would like to conclude with two quotes of two scientists. One is Dick Karp said that biology is now understood as information sciences, and scientists seek to describe biological processes as algorithms. In other words, nature is computing. On the other hand, John Kleinberg said that social networks are pre-technological creations that sociologists have been analyzing for decades, but we're now witnessing a revolution in measurement. Social networking research promises a rich trove for marketers and politicians, as well as sociologists, economists, anthropologists, psychologists, and educators, and I would say biologists. So this is an introduction of computing and algorithmic processes into the social sciences in a big way, and we're just at the beginning. So I would like to thank my graduate students and collaborators, Jared Say from the University of New Mexico, David Kempe from the University of Southern California, Dan Rubenstein, the head of Ecology Evolutionary Biology Department at Princeton, his postdocs, Ilya Fishoff and Shiva Sundaresan, who work in Kenya, and these generous funding sources that support our research. Thank you.
Okay, I think we have some time for some questions.
So in the assumption that you talked about where a few slides ago where you said that things are monotonic, an active person continues to be active.
Mm-hmm. Okay.
Previous slide maybe.
Yeah. Sorry, yeah.
Yeah.
Right.
Active nodes never deactivate.
Mm-hmm.
How realistic is that?
It's realistic because we can answer. No, so in epidemiology, it corresponds to the SIR model, susceptible, infected, recovered. Once you're recovered, you never again become infected. So it's actually pretty realistic for some diseases and for some.
They don't activate other nodes after they're I mean, there's a period where.
Right. So this you would really consider it for the period of infection. So it's for fast-spreading diseases that can cause epidemics, and where the incubation and when the infectious period itself is what you would consider basically. So they typically are considered for the 3 times the infectious period, right? So the three frames of the infectious period. So it's SIR model. It's pretty standard for things like smallpox or any other fast-spreading disease, where SARS. Where by the time you recovered, it spreads. You can infect so many people and it can spread so fast that all your intervention, if you really are serious about doing it, has to happen within that period before the recovery.
Yeah, but if you look at it across time, maybe I should take this offline, but it looks to me that in fact, there's a period in which your
infectious.
Your clusters may grow, and then other periods where they may.
Right. So for fast-spreading diseases, all intervention, and so one of the things that's been shown through simulations or other models is that early intervention is what matters. For example, that by the time, if your infectious period is 3 days, all intervention has to happen within that time for the population, not for individual. Yeah. But there are clearly also, there are kind of two classes of models in epidemic. There's SIR, susceptible, affected, recovered, and there's SIS, which is susceptible, infected, susceptible. So there's a cycle. Yeah. This is not SIS. Yes.
It seems that what ends up happening is, for this to be reasonable, we need the infectious period to be at least several multiples of the incubation period, because our time step has to correspond somewhat to the incubation period, the time in which I can turn a neighboring individual.
Right. So there are diseases for which incubation period is also infectious, and infectious in a different way. So we're not claiming here to build a realistic model of disease spread. We're taking sort of the standard disease spread model, the standard spread models, and seeing what effect do they have on the computational models of identifying these critical individuals. Whether the first question which we asked was, do we really need the dynamic model? Maybe it doesn't really matter. Maybe these aggregate models are doing a reasonably good job of identifying, of estimating what's going on in these networks. And well, actually, you can do a mental exercise of a simple network, which is, I showed at the beginning, a path, right? You can get arbitrary off between the dynamic and the static networks. You can misestimate, drastically what's going on in the static and the dynamic. So the next question is, well, for which kinds of networks does it matter? Can you look at the network and look at the structure of the network and say, well, for this network, because of its density, maybe average path length or whatever, it really doesn't matter. We really can just, go with the aggregate models, or even more than aggregate, this big sort of demographic models of differential equations, and we're going to get more or less the right information, because it's hard to collect this detailed information of dynamic networks. So, and then the next question is, okay, well, instead of doing these simulations, are there structural indicators of the individuals that are critical that we should focus on and not bother with everybody else? Or can you identify all of this as the sort of the process of spreading unfolds? How much time do you have to collect information? That sort of direction where we're going. But you're right. You need to be more realistic about it. You need to be more realistic about the actual model of disease spread, or the process that's spreading in the population. Yeah.
So coming back to the donkeys versus zebra, depending on which side of the Atlantic you're on.
We're evenly divided.
It would be extremely tempting to conclude that, well, the donkey populations have less structure to them because they sort of intermingle much more. How much can one be confident that actually this is some artifact of their environment that you're seeing here, and under the same conditions as the zebra population, you'd have similar structure? So I'm trying to determine how much the environmental factors may be disrupting what otherwise would be things that you can infer from the data.
Right. So by environmental factors, you may think the size of the territory, the connectedness between the male territories, the water hole availability, things like that.
Stuff that's going to be so idiosyncratic to whichever group
right.
you're looking at, that actually there isn't any pattern. There's a pattern for that group over this period, but in a different situation, the group would be completely different because the geography or the weather or some other factor would disrupt.
So there are two things to it. One, if you observe, first of all, they're pretty similar ecology for those two populations, which is one of the reasons why biologists focus on those two. There's very similar ecology. We also can control for some of it by having, so onagers exist in India and in Israel, and we have data. We're now going to compare onagers in India to onagers in Israel, so you can eliminate some of that environmental noise. It's not environmental noise, environmental factors. So you can also compare two different Grevi populations in Africa. One is in Kenya, the other one is south. I forgot how much south. Maybe, I don't remember how much south. But it all depends on the outcome of elections, right? So, unfortunately. So you can eliminate part of it. There's also, they've done some controlled experiments with Grevis by changing the waterhole location, which is what basically determines the territory of the males, is the waterhole. So they have these troughs that they build, and then they take away the water from the troughs and study the decision of how to go to another one. Because it's the females actually the ones who decide which next waterhole to go and which groups they're going to merge or not merge or join or whatever it is. So so there is it's some of it they can eliminate. But what the main, I think, power of this is not to answer the questions, but ask the right ones. It's to pose the hypothesis that, okay, well, from this, it looks like this species have much less loyalty, that the interaction are driven much less by loyalty, and there is less cohesive structure here than here. Now the biologist can go ahead and ask the question, why? What is it? And start isolating the factors and focusing on this. Because we have a strong evidence of the difference in their social interactions and persistence of their social structure and maybe personal loyalty.
Okay, well, please, thank you, Steve.
Thank you.